metastasis, and so on and so on. And we use these technologies that I will talk about now just to improve our models, to mouse models, to study these diseases. So, as I said, we work with mice. And when we talk about GMOs in animals, Richard has a lot talked about GMOs in plants. I will talk only about animals because my feeling is that they are more sensitive to the public opinion than plants. I will talk first about mice. There it all started. I will talk about transgenics and knockouts. And all the technology that is developed in mice is now being used also in other species. And fortunately, the other species are much more complicated to work with, and it takes a lot more time to be efficient. So we have a lot more time to think about it, what we are doing. So that's fortunate about these higher, larger animals. Now, knockouts and transgenics. What is a knockout? A knockout is a species that misses a gene or multiple genes. So it has gene loss, and you can have it conditionally and you can have it constitutive. Constitutive, all the cells miss that gene, and the gene is deleted already in the germline. A conditional knockout is a knockout mouse is a mouse which is at will knockout in a certain tissue, the brain, the liver, the blood, wherever, at a moment where you want to have it deleted there. That's a complicated technology, but it's possible. Transgenics, in our nomenclature, is a species, is a creature which has additional genetic information, and that can be multiple genes, that can be only one gene, combination, and so on. I have to have my glasses. So it can be a foreign gene, which is foreign to the species which you are inserting it. It has multiple copies, and you can have it expressed in other tissues than originally. You can take, for instance, hemoglobin, which is only present in blood, but you can also express it in the liver or in the brain. Scientifically, it doesn't make sense, but okay, you can do it. So, what kinds of questions do we want to answer making transgenic animals? First of all, you can just basically study gene regulation. So a gene consists of a structural, it's a linear information. It's just a linear molecule of DNA. And you have